Hello again, how's it going? In the previous video I mentioned that we're going to look at this topic this time around. So the issue is how many genes are there in the human genome. And even the title says the human genome contains at least 26,000 genes, but the exact gene number is difficult to determine. If you look at some of the notes, okay, this paper appeared in 1993 by Antikar and Bird. It says based on the number of CPG islands, and they estimated about 80,000 genes. We'll look at this paper later. Another paper by Feng Liang in the year 2000. This paper calculates, extrapolated from the number of ESTs. Oh, hang on. Before that, do you know what CPG islands are? CPG islands are a stretch of DNA in the human genome that contains a high number of C's and G's in juxtaposition. Why are they important? These are sites of methylation, and they appear in quite a number of genes, especially housekeeping genes. So what Antikara did was from results in the human genome sequencing, they estimated the number of CPG islands they found, and from the number of CPG islands they found, they extrapolated and says, okay, because they estimated about 56% of the genes have CPG islands, so the number of CPG islands are extrapolated to about 80,000 genes. Okay. Feng Liang in 2000 used calculations and looking at ESTs. ESTs are expressed sequence tags. What are expressed sequence tags? If you know what are sequence tag sites are unique sequences in the genome. You use it as a landmark throughout the whole genome, the whole of the human genome, from chromosome 1 to chromosome 22, X and Y. Right? They identify sequences that can be used as landmarks, and this has been known as STSs, or sequence tag sites. ESTs, on the other hand, are expressed sequence tags. Again, they are unique sequences, and while some of them have been mapped, others have not been mapped, but they have information about where the tissues come from. ESTs come from cDNA libraries. Sequencing of cDNA libraries, and cDNA comes from different sets of tissues. Sometimes you have tissue from the retina, the heart, the brain, so, and then from there you get the RNA, you do sequencing of the cDNA, so you get the ESTs. So Feng Liang and colleagues, based on the number of ESTs that they've deposited or ESTs that's been found in the databases, they extrapolated to approximately about 120,000 genes. Funnily, this is near 2000, in the same issue of Nature Genetics, or was it later? I think it's Nature Genetics. In the same issue, Brent Ewing and colleague says, using the same resources, looking at ESTs as well, he estimated only about 35,000 genes. So again, it's a question that the people are trying to answer ever since then, and I can tell you now they have not actually determined. A rough idea that comes from now every now and again is about 26,000. Even the book explains. I'll bring you to this paper from Fields. All right. Now we're not going to read through the paper. I'm just going to show you this box. It's quite interesting. I hope you can see this. It says, what counts as a gene? So counting genes require being clear about what counts as a gene. Genes is a notoriously slippery concept, and differing notions about what it means to identify one can lead to heated disagreements. Here in this paper, he mentions that the relationship between a genetic definition of a gene and the physical properties of a gene is not a simple correlation. It says Recent work suggests that many genes encoding small regulatory RNAs. Now, this paper is way back in the 1994. So already then they said, there are a lot of small regulatory RNAs that act directly on transcripts may exist. They're not very sure then, but yeah, they're very sure now. Um, and then it says transcription units may encode multiple unrelated proteins, not only in prokaryotes virus but also in C. elegans. This is talking about alternative splicing. Alternative splicing produces multiple variations of a given protein from a single transcript. Then transcription units may overlap. So when they overlap, what are you talking about? They are single genes or two genes. And what about methylation sites? In the face of this complexity, we have interpreted counting genes as counting distinct transcription units or parts of transcriptions that may be translated to generate one or a set of related amino acid sequence. We will go to this book back and go to page 275. I think I showed you this in class, and where it says revising the concept of the gene in the post-genome era. So this, bearing in mind that this is 2011, again, so initially, as I mentioned, when I studied this course, a gene is as simple as a green gene, a red gene, and a blue gene. And well, you can see genes on either side of the double-stranded DNA. But recent analysis have found several other regions that this is what they call as TARs, or transcriptionally active regions. You actually find a transcript that combines two different genes by adding sequences that were not there before. There are transcripts that you have one part of the gene. Again, it traverses other parts, quite far away from the gene. So again, it says blurring of the gene boundaries and transcript levels. So because of that, you don't really know how many genes there are, unfortunately.